Imagine standing on the surface of Mars. The sky above you is a pale, dusty salmon color, neither the deep blue of Earth nor the black of open space, but something in between, something alien, something that your brain instinctively knows is wrong. The sun is there, you can see it, but it looks smaller than you've ever seen it, a cold, distant coin of light that gives barely 60% of the warmth it would give you back home. The ground beneath your boots is red iron oxide, ancient rust, the oxidized corpse of a world that may once have had rivers and oceans and perhaps, just perhaps, the stirrings of life. The temperature right now, in what passes for a Martian afternoon at the equator, is around -20 degrees Celsius. But you know that tonight it will plunge to -70, maybe -80. You know that the atmospheric pressure around you is less than 1% of what your body evolved to breathe. You know that without your suit, the carbon dioxide atmosphere would asphyxiate you within minutes. You know that the radiation streaming through that thin sky, unfiltered by a magnetic field that died billions of years ago, is slowly, silently, invisibly damaging every cell in your body. And yet you are standing here, not in a dream, not in a simulation. You are here on Mars, and the question that haunts the entire species you represent is this: how long can you survive? Not with the suit, not with the habitat behind you, pressurized and warm and stocked with freeze-dried food and recycled water. How long can the human body, the biological machine that evolution spent 4 billion years building for a very specific planet, how long can it function here? And the deeper question, the one that scientists and geneticists and visionary billionaires and quiet researchers in laboratories from California to Beijing are actually asking right now, is not just how long you can survive, but whether we can change the human body itself, at the level of its DNA, so that it doesn't just survive on Mars, but actually thrives. That question, genetic engineering for space, modifying humans to survive Mars, is not science fiction anymore. It is one of the most serious, most controversial, most morally complex scientific conversations happening on Earth right now. And by the time the story is over, you will understand exactly why it might be the most important question our species has ever asked. Let's start at the beginning. Not the beginning of Mars exploration, though we'll get there. Let's start at the beginning of the human story, because understanding why Mars is so dangerous for us requires understanding what we actually are, biologically speaking. The human body is a masterpiece of evolutionary engineering, but it is a masterpiece built for one very specific environment. Think about it. For the last 300,000 years, Homo sapiens has lived on Earth. Before us, our hominid ancestors lived on Earth for millions of years. Every single physiological system we possess, our cardiovascular system, our immune system, our neurological architecture, our bone density mechanisms, our hormone regulation, our reproductive biology, every single one of these systems was shaped by the specific conditions of one planet. A planet with roughly 1 atmosphere of pressure. A planet with a magnetic field that deflects the vast majority of harmful cosmic radiation. A planet with an atmosphere composed of roughly 21% oxygen and 78% nitrogen. A planet with gravity at precisely 9.8 meters per second squared. A planet with liquid water, with a biosphere teeming with microorganisms that our immune systems co-evolved with over millions of years, with a 24-hour day-night cycle that our circadian rhythms are synchronized to with extraordinary precision. Mars has none of that. Mars is, in almost every measurable biological parameter, the opposite of what the human body needs. And the extraordinary thing, the thing that should make you stop and genuinely marvel, is that despite knowing all of this, our species is planning to go there anyway. And some of the most brilliant minds in biology, in genetics, in bioethics, and in astrobiology are now seriously asking whether, instead of building better suits and better habitats, we should be asking whether we can build better humans. To understand why genetic modification for Mars is even being discussed, we need to understand exactly how hostile Mars is, not in the abstract, but in the specific, visceral, biological detail that makes this whole conversation feel both urgent and slightly terrifying. Start with radiation. On Earth, we are protected by 2 overlapping systems. The first is the planet's magnetic field, the magnetosphere, which deflects a large percentage of the charged particles streaming from the sun, the solar wind, and the even more powerful cosmic rays streaming from distant supernovae and other galactic events. The second layer of protection is Earth's atmosphere, which absorbs much of the radiation that gets through the magnetosphere. On Earth, the average person receives about 3 millisieverts of radiation per year from all natural sources combined. For context, a single chest X-ray delivers about 0.1 millisieverts. A full-body CT scan delivers about 10. That's the kind of scale we're talking about. On Mars, it's completely different. NASA's Curiosity rover carried a radiation detection instrument called the Radiation Assessment Detector, and the data it sent back was sobering. A 6-month trip to Mars through interplanetary space would expose an astronaut to roughly 300 millisieverts of radiation. That's 100 times the annual limit for a radiation worker in the United States. That's from the journey alone, before you've even landed. Once on Mars, the surface radiation environment is significantly better than in space because you at least have the thin atmosphere and the ground-blocking radiation from below, but it's still approximately 10 to 20 times higher than on Earth's surface. Over the course of a 3-year mission, the cumulative radiation dose would increase a person's lifetime cancer risk by somewhere between 3 and 5%, according to NASA estimates, though some researchers think those figures may be underestimates when you account for the full spectrum of radiation effects. But cancer risk is only part of the story. Radiation doesn't just damage DNA in ways that might eventually cause cancer. It damages the central nervous system directly. Studies on astronauts who have spent extended time on the International Space Station, which is still within Earth's protective magnetosphere and therefore receives far less radiation than a Mars mission would, have already shown measurable changes in brain structure and cognitive function. A landmark study published in 2017 in Nature Microgravity found that astronauts who had spent 6 months on the ISS showed changes in white matter microstructure and altered cerebrospinal fluid distribution that persisted for years after their return. Now imagine that, but multiplied by the radiation environment of a Mars mission, which is orders of magnitude worse. There is a particular type of radiation that space agencies find most alarming: high-energy galactic cosmic rays, specifically the heavy ion particles called HZE particles, for high charge and energy. These are atomic nuclei, things like iron nuclei, carbon nuclei, that have been stripped of their electrons and are traveling at speeds close to the speed of light. When one of these particles passes through human tissue, it doesn't just nick a DNA strand. It tears through it. It creates a track of destruction through multiple cells simultaneously, like a bullet ripping through paper. And unlike X-rays, which human cells have evolved some capacity to repair, HZE particle damage is often irreparable or repaired incorrectly, and the biological consequences at the cellular level are still not fully understood. Research published in 2018 in Scientific Reports from the University of California, Irvine showed that mice exposed to charged particle radiation similar to galactic cosmic rays experienced significant impairments in learning and memory, increased anxiety, and neuroinflammation. These were not effects that faded after the exposure ended. They persisted. They were, in effect, a kind of accelerated neurological aging. So here is a question I want you to sit with for a moment. If you were designing a human being from scratch, if you could edit the blueprint of the human genome to prepare a person for Mars, what would you change first? Would it be the radiation resistance? The ability to breathe a different atmosphere? The physiology to cope with lower gravity? The gut microbiome to function without Earth's ecosystems? Think about that. Think about where you would start, because it tells you something about how you think about what it means to be human. Let's move on to gravity, or rather, the lack of it, or more precisely, the wrong amount of it. Mars has about 38% of Earth's surface gravity. That means if you weigh 70 kilograms on Earth, you weigh about 26.5 kilograms on Mars. That sounds wonderful, and in some ways it is. You can jump higher, carry more, move with less effort. But your body, the biological machine built over millions of years for Earth's gravity, does not think it's wonderful. It thinks something has gone catastrophically wrong. What happens to the human body in microgravity, as we know from the International Space Station, is a kind of slow-motion deterioration. Bone density drops. In microgravity, the body loses about 1 to 2% of bone mass per month in load-bearing bones. After 6 months in space, a significant portion of astronauts have bone density comparable to someone decades older than their actual age. Muscle mass decreases at a similar rate. The heart, which normally works against gravity to pump blood upward to the brain, suddenly doesn't have to work as hard. It gets lazy. It atrophies. The cardiovascular system recalibrates for an environment without gravity, and this recalibration has serious consequences. Fluids shift upward toward the head, causing what astronauts describe as a permanent head cold feeling, congestion, facial puffiness, and increased intracranial pressure that can compress the optic nerve and impair vision. This vision problem, known as spaceflight-associated neuroocular syndrome, or SANS, has affected a remarkable proportion of NASA astronauts. A study published in JAMA Ophthalmology found that about 40% of astronauts who completed long-duration ISS missions reported vision problems, some of which persisted after return to Earth. Now, Mars is not microgravity. It's partial gravity, 38% of Earth's. The question of how the human body responds to long-term exposure to Martian gravity specifically is something we simply don't know yet, because we've never had humans in partial gravity environments for extended periods. The moon landings were too brief. The ISS is microgravity. We have no data on what living at 0.38 G does to bones, muscles, the cardiovascular system, and vision over years or decades. But the reasonable scientific assumption, based on what we know about microgravity, is that it would be somewhere between zero gravity and Earth gravity, which is to say that it would still cause significant physiological deterioration over time, just more slowly. Bone loss would likely still occur. Muscle atrophy would still be a problem. Whether the cardiovascular and vision issues associated with microgravity would manifest to the same degree in partial gravity is genuinely unknown. And here is the terrifying part. If you spend years living on Mars, adapting your body to 0.38 G, and then you try to come back to Earth, the readaptation to Earth's full gravity might be devastating. Bones that have lost density might fracture. Muscles that have atrophied might be insufficient to move a body that is suddenly nearly 3 times heavier than it was on Mars. A heart that has recalibrated for partial gravity might struggle. The first generation of long-term Mars residents might be functionally unable to return to Earth. They might, in a very real sense, be committed to Mars from the moment they adapt. Does that scare you? It should, a little. But it should also make you wonder, because there is a certain beautiful, terrible logic to the idea that the first true Martians might not be people who went to Mars and came back. They might be people who went to Mars and stayed. And if we're going to design people for Mars, we wouldn't just be designing them to survive the journey. We'd be designing them to live there, maybe to never come back. What does it mean to engineer a human being for a one-way destination? Now let's talk about atmosphere, or rather, the absence of a breathable one. Mars's atmosphere is approximately 95% carbon dioxide, 2.6% nitrogen, 1.9% argon, and trace amounts of oxygen and other gases. The atmospheric pressure at the Martian surface is about 0.6% of Earth's sea level pressure. To put that in perspective, even if you could somehow breathe carbon dioxide, the atmospheric pressure on Mars is so low that it would be below the Armstrong limit, the pressure below which water boils at body temperature. This means that at Martian surface pressure, the fluids in your body would begin to vaporize. Your saliva would boil. The water in your tissues would turn to gas. This is called ebulism, and it is an extraordinarily unpleasant way to die, though you would lose consciousness in about 15 seconds from oxygen deprivation, long before the full effects of ebulism manifested. In any case, you would not survive unprotected on the Martian surface for more than a few minutes, likely less. This is why every serious Mars colonization plan involves either pressurized habitats that maintain Earth-like atmospheric conditions inside or spacesuits that do the same thing around the individual. But what if you didn't need that? What if you could engineer humans who could breathe, or at least tolerate, a radically different atmospheric composition? What if you could engineer a human whose hemoglobin could extract oxygen from an atmosphere where it's a trace gas? What if you could engineer a human who could sustain life at lower pressures? These are not idle science fiction questions. They are, with varying degrees of seriousness, being discussed in actual scientific literature. And they connect to one of the most extraordinary sources of biological inspiration we have, the diversity of life on Earth itself. Because here's the thing about life on Earth. It is shockingly, almost defiantly diverse in the environments it can tolerate. We call the organisms that thrive in extreme environments extremophiles, and their existence is one of the most important and underappreciated facts in all of biology. Let me walk you through some of them, because they are directly relevant to the genetic engineering conversation. Tardigrades, also known as water bears or moss piglets, are microscopic animals about 0.5 millimeter long that can survive conditions that would instantly kill any human. They can tolerate temperatures ranging from -272 degrees Celsius, just 1 degree above absolute zero, to plus 150 degrees. They can survive pressures 6 times greater than the deepest ocean trench. They can withstand radiation doses of up to 570,000 roentgens, a dose that would kill a human at about 500. And here's the most remarkable thing. They can survive in the vacuum of space itself. An experiment in 2007, part of the Photon M3 mission, sent tardigrades into open space attached to the outside of the spacecraft. They survived 10 days of exposure to vacuum and cosmic radiation and returned alive. How? They go into a state called cryptobiosis, essentially shutting down all metabolic processes, replacing the water in their cells with a sugar called trehalose that prevents ice crystal formation and protects cellular structures. Their DNA is also protected by a unique protein called SUP, for damage suppressor, that physically wraps around DNA and shields it from radiation damage. When researchers inserted the gene for SUP into human cells in culture, the human cells showed 40% greater resistance to X-ray radiation. Read that again. A gene from a tardigrade, when inserted into human cells, reduced X-ray-induced DNA damage by 40%. That experiment, published in 2016 in Nature Communications by a team from the University of Tokyo, is one of the most cited pieces of evidence in discussions about genetically engineering humans for radiation resistance in space. It is not a theoretical possibility. It has been demonstrated in cell culture. The gene works in human cells. The question is whether it could work in an actual living human being, in a whole organism context, without unintended consequences. Let's talk about other extremophiles. Deinococcus radiodurans is a bacterium that holds the world record for radiation resistance. It can survive doses of 1.5 million rads of ionizing radiation, which is 3,000 times the lethal dose for humans. After being blasted with radiation that would destroy most living things, Deinococcus radiodurans reassembles its shattered genome from fragments, like the world's most dedicated puzzle solver. It does this through an extraordinarily efficient DNA repair system, one that can reassemble hundreds of double-strand DNA breaks simultaneously without significant error. The mechanisms behind this ability are being studied intensively, and some researchers believe that elements of this DNA repair pathway could potentially be introduced into human cells to enhance our own radiation tolerance. There are also organisms that thrive at the low pressures and low temperatures of high altitudes, animals that have evolved to function at oxygen levels far below what would incapacitate most humans. The Tibetan people are perhaps the most famous example of human evolution for environmental adaptation. Tibetans have lived at altitudes above 4,000 meters for at least 15,000 years, and during that time they have evolved specific genetic adaptations that allow them to function normally in the low oxygen conditions found at high altitude. The most important of these adaptations is in the EPAS1 gene, which regulates hemoglobin production and oxygen utilization. In most humans, low oxygen levels trigger overproduction of red blood cells, which thickens the blood and increases the risk of stroke and blood clots. Tibetans, through natural selection acting on variants of EPAS1, have evolved a different response. Their hemoglobin has a higher affinity for oxygen, allowing them to extract more oxygen from thin air without overproducing red blood cells. This genetic adaptation is so unusual that researchers have traced it back to a population of ancient humans, the Denisovans, from whom Tibetans appear to have inherited the variant through interbreeding tens of thousands of years ago. The Denisovan connection is important, and it's beautiful because it shows that this kind of genetic modification for environmental adaptation has already happened in human evolutionary history. We have already borrowed genes from another population to adapt to a new environment. What genetic engineering for Mars proposes to do is the same thing, but deliberately and rapidly, instead of waiting hundreds of generations for natural selection to do it. This brings us to the actual science of genetic engineering, because we need to understand what our tools are before we can understand what they could theoretically do. The conversation about genetic engineering for Mars is inseparable from the development of a specific technology that has, over the past decade, transformed biology more profoundly than anything since the discovery of the structure of DNA. That technology is CRISPR-Cas9. CRISPR, which stands for Clustered Regularly Interspaced Short Palindromic Repeats, is a bacterial immune system. In nature, bacteria use CRISPR sequences to remember and defend against viruses they've previously been infected by. When they encounter a virus a second time, molecular machinery called Cas proteins uses the CRISPR sequences as a guide to find and cut the virus's DNA, disabling it. In 2012, Jennifer Doudna at the University of California, Berkeley, and Emmanuel Charpentier, then at Umeå University in Sweden, published a landmark paper in Science demonstrating that this bacterial system could be reprogrammed to cut DNA at any sequence you chose. You could design a guide RNA that matched any genetic sequence you wanted, introduce it into a cell along with the Cas9 cutting protein, and the system would find that exact sequence in the genome and make a precise cut. This was an almost incomprehensibly powerful discovery. It meant that any gene in any organism could potentially be edited, deleted, modified, or replaced with a precision and ease that made all previous genetic engineering techniques look like cave painting compared to digital art. Doudna and Charpentier won the Nobel Prize in Chemistry in 2020 for this work. In the decades since their original paper, CRISPR technology has been used to cure sickle cell disease in human patients, to develop new cancer therapies, to create disease-resistant crops, to resurrect traits from extinct organisms, and to generate animals with human organs for transplantation research. The technology has moved from laboratory curiosity to medical intervention with breathtaking speed. And in 2018, it crossed a line that shocked the world. In November 2018, a Chinese biophysicist named He Jiankui announced that he had used CRISPR to edit the germline, meaning the embryonic cells that would eventually become every cell in the organism, including eggs and sperm, and therefore could be inherited by future generations of human embryos. Two baby girls, twins he called Lulu and Nana, had been born from those embryos. He Jiankui had edited their CCR5 gene, which encodes a protein that the HIV virus uses to enter cells. By disabling CCR5, he claimed to have made the children resistant to HIV infection, following a precedent set by the natural CCR5 delta 32 mutation found in some individuals of European ancestry who are naturally resistant to HIV. The scientific community was horrified. Not because the technical achievement wasn't real. It apparently was, though the work has never been independently peer-reviewed. They were horrified because He Jiankui had edited the germline of human beings, changes that would be passed to future generations, without adequate safety testing, without proper ethical review, and without the informed consent of the children who would carry these changes forever. He Jiankui was subsequently convicted of illegal medical practice by a Chinese court and sentenced to 3 years in prison. But the genie was out of the bottle. The first genetically edited human beings were already alive. The question of whether and how we should engineer human genetics was no longer purely theoretical. The He Jiankui case also revealed something important about the CCR5 gene specifically, something that bears directly on the Mars conversation. Research published after his announcement showed that the CCR5 delta 32 mutation, the same deletion he had tried to engineer, is associated with increased susceptibility to certain other infections, including West Nile virus, and also, intriguingly, with increased risk of premature death according to a large population study. Genes are rarely simple switches with one function. They are embedded in complex networks of biological interactions, and changing one gene can have cascading, unpredictable effects throughout the entire system. This is called pleiotropy, and it is one of the central challenges of genetic engineering for human enhancement. You might fix one problem and inadvertently create several others. But let's get back to Mars, because the He Jiankui case, as disturbing as it was, represents only the most primitive beginning of what would be required to genetically engineer humans for Martian survival. We're not talking about editing one gene to confer resistance to one virus. We're potentially talking about editing dozens or hundreds of genes across multiple biological systems to create a human being who can tolerate a radiation environment 10 to 20 times more intense than Earth, a gravitational field 38% of Earth's, an atmospheric composition that is nearly unbreathable, average temperatures that would be lethal without thermal protection, a day length of about 24 hours and 37 minutes, which is actually close enough to Earth's that circadian rhythms might adapt reasonably well, one of the few things Mars is going for it biologically, and a complete absence of the terrestrial microbiome that human immune systems co-evolved with. Let's go through these one by one, exploring what genetic engineering might theoretically do about each of them, what the actual science says, and what would need to happen to make any of it a reality. Radiation resistance first, because it's probably the most urgent biological challenge for Mars colonists, and because there's actually the most scientific discussion about it. The human body has sophisticated DNA repair mechanisms already. We didn't evolve in a radiation-free environment. Earth's surface has always had background radiation, and our cells are constantly repairing radiation-induced DNA damage. But our repair mechanisms are calibrated for Earth's background radiation levels. They are woefully insufficient for the radiation environment of a Mars mission. So what would we need to add or change? The first target, based on existing research, would be the DNA damage response pathway, the complex network of proteins that detects DNA damage and orchestrates the repair response. Researchers have identified specific proteins in organisms like Deinococcus radiodurans that are vastly more efficient at this than our equivalents. The RICA protein in Deinococcus, for example, is far more efficient at finding and repairing homologous sequences during double-strand break repair than our equivalent human protein, RAD51. Could we engineer enhanced versions of these repair proteins into human cells? Could we introduce the SUP protein from tardigrades, the DNA damage suppressor we talked about earlier, into human cells in a way that functions throughout the body? Then there's the antioxidant response. A large portion of radiation damage occurs not directly, through the radiation particles themselves, but indirectly, through the production of reactive oxygen species, free radicals generated when radiation ionizes water in cells. These free radicals then damage nearby molecules, including DNA, proteins, and lipids. Our cells have antioxidant defense systems, including enzymes like superoxide dismutase, catalase, and glutathione peroxidase, but they're not designed for the intensity of free radical production that would occur in the Martian radiation environment. Enhancing these antioxidant defenses, either by upregulating existing enzymes or introducing more powerful variants from radiation-resistant organisms, is another potential approach. There are also more radical proposals, and I mean radical in both senses of the word. One idea that has been discussed in the scientific literature involves engineering cells to be better at apoptosis, programmed cell death, specifically in response to severe radiation damage. The logic is counterintuitive. Instead of trying to repair badly damaged cells, you engineer cells to more readily sacrifice themselves when they detect irreparable damage, before they can become cancerous. This is essentially engineering a more aggressive internal cancer prevention system, accepting that some cells will die while ensuring that fatally damaged ones don't stick around to cause problems. Another approach involves engineering human cells to be more similar to the cells of naked mole rats, which have extraordinary cancer resistance. Naked mole rats live up to 30 years in the wild, about 10 times longer than mice of similar size, and they almost never get cancer. Researchers have discovered that this is partly due to a substance called high molecular weight hyaluronan, which their cells produce in unusually large amounts and which appears to trigger a cellular response called early contact inhibition, stopping cell division before tumors can form. They also have uniquely accurate protein quality control systems and unusually efficient DNA repair. If the cancer resistance mechanisms of naked mole rats could be understood and translated into human biology, it could be transformative not just for space but for human health on Earth. But here is the thing that makes all of this so extraordinarily complex. Radiation resistance in organisms like Deinococcus radiodurans and tardigrades works in part because those are simple organisms. Deinococcus is a single cell. Tardigrades have less than 1,000 cells in total. The human body has approximately 37 trillion cells, organized into more than 200 different cell types, arranged in tissues and organs with exquisitely coordinated functions. Engineering radiation resistance into a simple organism is a very different proposition from engineering it into a human being, where the changes have to work in every cell type, have to not disrupt the thousands of other biological processes those cells are involved in, and have to function across the entire lifespan. The more cells you have, the more complex your biology, the more potential sites for things to go wrong when you start changing the genetic code. This is why some researchers argue that the right approach isn't to engineer human beings for Mars at all. It's to engineer something simpler, and then use that simpler thing as an interface between humans and Mars. Synthetic biology, the design of biological systems that don't exist in nature, is another area that feeds into the Mars conversation. Instead of modifying humans, why not create synthetic organisms, perhaps derived from known extremophiles, that can help terraform Mars, creating a more hospitable environment over centuries? Or why not create synthetic organs, artificial biological systems that can be implanted into humans to augment specific capabilities, like a radiation-filtering artificial skin layer, or an enhanced oxygen extraction system in the lungs? These ideas blur the line between genetic engineering and cybernetics, between biology and technology, and that blurring is itself philosophically significant. When does modifying a human being stop making that being human? That question is not rhetorical. It is the central philosophical challenge of this entire conversation, and it's being actively debated right now in bioethics journals, in philosophy departments, in NASA advisory committees, and in the pages of scientific journals ranging from Nature Biotechnology to Acta Astronautica. Let's sit with it for a moment, because it deserves serious consideration. The idea that humanity has a fixed, immutable nature that should not be tampered with is a concept with deep roots in philosophy, religion, and common human intuition. There is something that feels sacred about the human genome, about the biological inheritance of 4 billion years of evolution. When He Jiankui edited the genes of Lulu and Nana, the reaction wasn't just scientific concern. It was visceral moral revulsion from people across the political and cultural spectrum. Something felt violated. But why? Philosophers distinguish between two broad positions on this question. Bioconservatives argue that there are intrinsic values to natural human biology, that our genome is not merely a technical document but something that embeds our humanity itself, and that altering it for purposes beyond the treatment of specific diseases crosses a line that could not be uncrossed. Transhumanists, on the other hand, argue that there is nothing sacred about our current biological form, that evolution has already been tinkering with us continuously for millions of years, and that adding our own intentional tinkering to that process is not categorically different, just faster and more deliberate. They argue that if genetic modification can reduce suffering, extend life, and expand human capabilities, then refusing to do it is itself a moral choice with moral consequences. The Mars context adds another layer to this debate. If the modification is not to enhance humans beyond their natural capabilities in the competitive context of life on Earth, but specifically to enable survival in an environment where unmodified humans simply cannot survive, does that change the moral calculus? If you are going to Mars and you will not survive without genetic modifications, is choosing those modifications really so different from choosing to wear a spacesuit? One is a technological solution and the other is a biological one, but both serve the same purpose: enabling human survival in an environment we didn't evolve for. There's a thought experiment that I want you to consider right now, wherever you're listening to this. Imagine that you have a child, and you have the opportunity, before that child is born, to edit 3 of their genes. The first edit would give them dramatically enhanced radiation resistance, making them far less likely to develop cancer from radiation exposure throughout their life. The second would give them enhanced bone density maintenance, making them less susceptible to osteoporosis as they age. The third would give them enhanced oxygen utilization efficiency, allowing them to function better at high altitudes and in low oxygen environments. None of these changes would affect their appearance, their intelligence, their personality, or their social relationships. They would simply be healthier, more physically resilient. Would you do it? Most people, when asked this question in studies on public attitudes toward genetic enhancement, say they would. And here's the thing. Those 3 edits I just described are exactly the kind of edits that would be relevant for a Mars mission. The line between therapeutic genetic modification, which most people are willing to accept, and enhancement for space survival, is blurrier than we think. Now let's talk about the microbiome, because this is one of the most fascinating and underappreciated aspects of the genetic engineering for Mars conversation. The human gut microbiome, the complex ecosystem of trillions of bacteria, viruses, fungi, and other microorganisms that live in our digestive tract, is not just a passenger in our bodies. It is an integral part of our biology. The gut microbiome helps us digest food, synthesize vitamins, regulate our immune system, and even influence our mood and cognitive function through what scientists call the gut-brain axis. Research over the past 2 decades has fundamentally transformed how we think about human biology. We are not individuals. We are ecosystems. The problem is that the gut microbiome of a Martian colonist would be radically disrupted by the space environment. Multiple studies of ISS astronauts have shown significant changes in gut microbiome composition during spaceflight. Research published in 2019 in the journal Science found that the gut microbiomes of twin astronaut Scott Kelly, who spent a year on the ISS, and his identical twin brother Mark Kelly, who stayed on Earth, diverged significantly during Scott's mission and did not fully return to their pre-mission state even months after his return. The loss of diversity in the gut microbiome, and the specific changes in which species are dominant, have wide-ranging health implications that we are only beginning to understand. On Mars, the situation would be even more extreme. The radiation environment would be highly damaging to gut microbiome bacteria, which are not radiation resistant. The food supply on Mars, at least initially, would be radically different from an Earth diet, likely consisting largely of hydroponically grown plants, cultured proteins, and highly processed preserved foods, which would further alter microbiome composition. The complete absence of the terrestrial microorganic environment that humans evolved with would mean that the gut microbiome of Mars colonists would, over generations, become something very different from anything found on Earth. Some researchers argue that deliberately engineering the human gut microbiome for Mars might be as important as engineering the human genome itself. The idea would be to design a suite of microorganisms specifically selected or engineered for stability in the Martian diet environment, for radiation resistance, for the production of specific vitamins and nutrients that might be absent from a Martian diet, and perhaps even for functions not found in any terrestrial microbiome, like the production of compounds that help repair radiation-damaged tissue. This engineered microbiome would be colonized deliberately in Mars-bound humans, essentially replacing their natural microbiome with one specifically designed for their new environment. This is a form of biological engineering, though it operates at the ecosystem level rather than the individual genetic level. And it raises its own profound questions. If your gut bacteria are engineered specifically for an alien environment, does that make you less human? Does it matter? And what happens if you try to return to Earth with a microbiome that has been optimized for Mars? Would you even be able to digest Earth food? Would your immune system, co-evolved with your engineered microbiome, be able to cope with Earth's vastly different microbial environment? Let's shift from biology to the actual organizational and institutional conversation about genetic engineering for Mars, because it's important to understand that this is not just happening in academic journals. It is being discussed at the highest levels of space exploration and biotechnology. The most prominent advocate for human Mars colonization is, of course, Elon Musk and his company SpaceX. Musk has been explicit that he believes humanity must become a multi-planetary species to ensure long-term survival, and Mars is his chosen destination. SpaceX has developed the Starship rocket specifically with Mars colonization in mind, and Musk has spoken about wanting to establish a self-sustaining city of 1 million people on Mars within the next few decades. However, Musk himself has been relatively quiet about the specific biological challenge of keeping those 1 million people healthy, focusing more on the engineering challenges of the rocket and the habitat. When pressed on the radiation issue specifically, his responses have been somewhat dismissive, suggesting that it's a manageable risk, comparable to what he calls the background risk that everyone faces on Earth from living life. Most radiation biologists would strongly disagree with that assessment. NASA's approach is more conservative and more scientifically rigorous. NASA has been studying the health effects of spaceflight intensively, particularly since the long-duration mission programs on the ISS. The NASA Human Research Program has identified 5 specific risk areas for long-duration spaceflight, which they call the 5 hazards: radiation, isolation and confinement, distance from Earth, gravity fields, and hostile or closed environments. They have spent hundreds of millions of dollars studying these risks and developing countermeasures. However, NASA's current countermeasure strategy is focused almost entirely on engineering and medical solutions. Better shielding, exercise protocols, pharmacological interventions, not genetic ones. The idea of genetically engineering astronauts for Mars is not part of NASA's current roadmap, but it is part of the conversation at organizations like the NASA Innovative Advanced Concepts Program, which funds early-stage speculative research that could eventually lead to breakthroughs. And it is being seriously explored by researchers at institutions including Harvard, MIT, UC Berkeley, and the J. Craig Venter Institute, the genomics powerhouse founded by one of the scientists who led the Human Genome Project. Craig Venter himself, who in 2010 created the first synthetic organism with a computer-designed genome, a bacterium called Mycoplasma mycoides JCVI Syn 1.0, has spoken about the potential for synthetic biology to solve problems in space colonization. The idea of designing organisms from the genetic level up, rather than editing existing organisms, represents a different and in some ways even more radical approach than CRISPR. Instead of modifying the human genome, you might design biological machines, synthetic cells or organisms, that perform specific functions humans need on Mars. One of the most remarkable and often overlooked figures in the genetic engineering for Mars conversation is Christopher Mason, a geneticist at Weill Cornell Medicine in New York City. Mason is the author of a book called The Next 500 Years, Engineering Life to Reach New Worlds, published in 2021, and he has been one of the most prominent and serious voices arguing that genetic engineering for space is not just possible but necessary. His argument is essentially a moral one. If we are going to send people to environments that will harm them, we have an obligation to do everything we can to protect them, and genetic engineering is one of the tools available to us. To refuse to use it on grounds of squeamishness while sending people to their biological detriment is itself an ethical failure. Mason has been involved in research that has taken some extraordinary approaches to understanding the genetic basis of adaptation to space. He was a co-investigator on NASA's Twin Study, the comprehensive scientific study of Scott Kelly and his twin brother Mark that we mentioned earlier. The Twin Study, published in 2019 across multiple papers in the journal Science, revealed some stunning findings about the effects of spaceflight on human biology at the genetic level. Scott Kelly's gene expression patterns changed significantly during his year in space. Thousands of genes changed their activity levels, with particularly notable changes in genes related to the immune system, the DNA damage response, bone formation, hypoxia response, and even cognitive function. Most of these changes reversed within 6 months of Scott Kelly's return to Earth, suggesting they were adaptive responses rather than permanent damage. But about 9% of the changes in gene expression did not fully reverse during the follow-up period, suggesting either lasting adaptation or lasting damage. Perhaps most startling was the finding that the telomeres, The protective caps on the ends of chromosomes that shorten with age and stress actually got longer in Scott Kelly's white blood cells during his year in space, only to rapidly shorten again upon his return to Earth. This was unexpected and counterintuitive, though researchers caution that longer telomeres are not always a good thing, as certain cancers are associated with abnormally long telomeres. The twin study showed clearly that the human body is not a passive victim of the space environment. It responds to that environment dynamically, changing its gene expression patterns to cope. The question is whether those adaptive changes are sufficient, whether they go in the right direction, and whether we could engineer a starting genetic baseline that made those adaptations more effective and less damaging. Mason's work at Cornell has also included research into the creation of what he calls a genetic toolkit for space, a set of potential genetic modifications, drawn from extremophile organisms and from natural human genetic variation, that could theoretically be introduced into the genomes of space travelers or colonists to improve their survival odds. He is careful to point out that his research is far from clinical application, and that significant ethical and safety questions would need to be resolved before any of this could happen in humans. But he is equally clear that the conversation needs to happen now, before we actually send people to Mars, rather than after. This brings us to one of the most important and emotionally resonant aspects of this whole story. The question of consent, specifically the consent of future generations. Here's the problem. If we engineer an embryo's genome so that the resulting person is better adapted to Mars, that person cannot consent to those changes. They will carry them for their entire life. They will pass them to their children. And if the changes were made in the germline, as Yi Jiankui's were, they will propagate through the entire lineage of that family forever, unless specifically reversed by future genetic engineering. The children of Mars colonists, who were never consulted, who never had the chance to say yes or no, would be born as a different kind of human being, one engineered for a different world. There are echoes here of some deeply troubling historical precedents. The history of eugenics, the early 20th century movement that sought to improve the human species through selective breeding and which was used to justify forced sterilizations, and, at its most horrific extreme, the Holocaust, casts a long shadow over any discussion of deliberate human genetic modification. The fear that genetic engineering for space could become genetic engineering for superiority, that the idea of designing humans for one environment could slide into designing humans for economic or social or intellectual performance more generally, is not irrational. It's rooted in real historical atrocities. And it's part of why the bioethics community is so vigilant about where these lines are drawn. But there is a counterargument that is morally powerful. Parents already make decisions for their children that those children cannot consent to. We choose where they are born, how they are raised, what vaccines they receive, what education they get. All of these choices shape who they become. If a couple is planning to raise their family on Mars, and they have the option to give their future children genetic modifications that will significantly reduce their lifetime risk of radiation-induced cancer, is choosing that modification so different from choosing to vaccinate them against disease? Is not choosing it, knowing that the Mars radiation environment will expose them to significantly elevated cancer risk, itself a form of harm? This is genuinely hard. The bioethics literature doesn't have clean answers here. What it does have is a growing consensus that the conversation needs to be public, inclusive, and conducted before the technology is deployed rather than after, which is exactly the pattern we failed to follow with Yi Jiankui. There's another dimension to this question that is rarely discussed, but is perhaps the most philosophically interesting. If we genetically engineer humans for Mars over multiple generations, if the Mars colony selects for individuals with specific genetic traits that confer Martian adaptation, if over centuries the Martian human population diverges genetically from Earth humans, what happens? Could Martians and Earthlings eventually become reproductively incompatible? Could we, through the intentional and unintentional forces of selection on Mars, be witnessing the beginning of human speciation? The birth, in geological real time rather than evolutionary time, of a new species of the genus Homo? This is not idle speculation. This is a question that evolutionary biologists take seriously. The philosopher and scientist J.B.S. Haldane, one of the founders of the modern evolutionary synthesis, speculated in the 1960s about what he called the first men in space becoming the last men in space, meaning that the selection pressures of space would inevitably create new human lineages. More recently, researchers like John Hawks at the University of Wisconsin have written about how rapid environmental change can drive rapid evolutionary divergence, even within a few hundred generations. If Mars colonists are genetically engineered and then further shaped by natural selection in the Martian environment for 500 or 1,000 years, the resulting population might be as genetically different from contemporary Earth humans as we are from Homo heidelbergensis, our archaic ancestor of 300,000 years ago. Now let's ask you something directly. How do you feel about that? Somewhere in the next 100 to 500 years, assuming the technology and the will both exist, there may be born on Mars the first human being who is more Martian than Earthling. Not metaphorically, genetically, biologically. A being whose DNA was designed, at least in part, for that red, cold, irradiated world. A being who might find Earth's gravity crushing, Earth's atmospheric pressure uncomfortable, Earth's microbiome overwhelming. A being who would look up at the night sky and see a tiny blue dot and know that it was where their ancestors came from, the way you might look at the African savanna and know that is where yours came from. How does that make you feel? Does it fill you with wonder? With dread? With both? Because it should. This is one of the moments in human history, and we are living in it right now, when the choices being made in laboratories and boardrooms and policy discussions are shaping something that will define our species for thousands of years. And most people don't know it's happening. Let's talk about some of the specific genes and biological systems that scientists are actually discussing in the context of Mars adaptation, because the specificity is important for understanding just how real and how detailed this conversation is. One area of intense interest is the p53 gene, often called the guardian of the genome. p53 is a tumor suppressor gene that plays a central role in the cellular response to DNA damage. When a cell's DNA is damaged, p53 is activated and it makes a decision. Can this damage be repaired, or is the damage so severe that the cell should destroy itself? If p53 chooses repair, it halts the cell cycle to give the repair machinery time to work. If it chooses destruction, it triggers apoptosis, the programmed cell death pathway. This decision-making role makes p53 one of the most important cancer prevention mechanisms in the body. p53 mutations are found in more than 50% of all human cancers. But here's the extraordinary thing. Elephants have 20 copies of the p53 gene, compared to the 2 copies found in humans. And elephants, despite their much larger body mass and much longer lifespan than most mammals, have remarkably low rates of cancer. This was first noted by statistician Richard Peto in the 1970s, and became known as Peto's paradox. You would expect larger, longer-lived animals to have higher cancer rates because they have more cells and more time for those cells to acquire cancer-causing mutations. But they don't. Elephants have 20 copies of p53 in their genome and appear to use them to aggressively trigger apoptosis in response to DNA damage, essentially sacrificing damaged cells before they can become cancerous. Research published in 2015 in JAMA and in Cell Reports demonstrated that elephant cells in culture do respond to radiation and other DNA-damaging agents with much higher rates of apoptosis than human cells. The theoretical implication for space is obvious. If you could engineer human cells to have multiple copies of p53 or enhance p53 activity, you might dramatically increase radiation-induced cancer resistance. Some researchers have proposed exactly this. But there's a catch, and it's a significant one. p53 is not just about cancer prevention. It's also involved in aging. There is evidence that very high p53 activity accelerates certain aspects of aging, possibly because the aggressive elimination of damaged cells depletes tissue stem cell populations over time. Engineering very high p53 activity might protect against cancer while accelerating other aspects of biological aging. The balance between cancer resistance and longevity is one of the central unsolved problems in the biology of aging. This is the dilemma of pleiotropy writ large. You pull one thread, and 3 others come loose. Another gene family of interest for the Mars context is the NRF2 pathway. NRF2 is a transcription factor, a protein that regulates the expression of other genes, and it is the master regulator of the cellular antioxidant response. When cells are under oxidative stress, which is exactly what happens during radiation exposure, NRF2 is activated and it turns on a whole cascade of protective genes, including those encoding superoxide dismutase, catalase, glutathione-related enzymes, and many others. There is significant interest in enhancing the NRF2 pathway in the context of radiation protection, and preliminary research in animal models has been promising. Some studies have found that animals with enhanced NRF2 activity show improved survival after radiation exposure, though the picture is complicated by the fact that NRF2 upregulation also increases cancer risk through a different mechanism, by helping cancer cells resist the oxidative stress that would otherwise kill them. Again, the biology resists simplification. Then there's the question of the skeletal system and the challenges of low gravity. The key molecular pathway governing bone density is a signaling cascade involving proteins, including sclerostin, encoded by the SOST gene. Sclerostin is produced by bone cells called osteocytes, and it acts as a brake on bone formation, preventing bones from becoming too dense. In low gravity, bones don't receive the mechanical loading signals they need to maintain density, and the sclerostin pathway essentially tells the body that the bones are more than adequate for the low-stress environment. The result is the bone loss we've already discussed. One approach to counteracting this would be to engineer reduced sclerostin function in Mars colonists, essentially making the brake on bone formation less responsive to the signals of low mechanical loading. There are already drugs that target sclerostin, including romosozumab, approved by the FDA in 2019 for treating osteoporosis in postmenopausal women. The existence of an approved drug targeting sclerostin suggests that the pathway is a valid therapeutic target, though a genetic modification approach would be more permanent and more broadly effective than a drug. Similarly, the muscle wasting that occurs in low gravity involves the myostatin pathway. Myostatin, encoded by the MSTN gene, is a protein that limits muscle growth. Natural mutations that inactivate myostatin in animals, including cattle, sheep, and even one documented human case, result in extraordinary muscle development. The so-called Mighty Mouse experiments, in which mice with inactivated myostatin genes developed muscles roughly twice the normal size, demonstrated proof of concept decades ago. A child born in Germany in 2004 with a natural myostatin mutation had muscle development at birth that was described as extraordinary, with visible muscle definition, and subsequent follow-up showed that the child maintained unusual muscle mass without specific exercise. Reducing myostatin activity in Mars colonists could theoretically help counteract muscle atrophy in the low-gravity environment, helping them maintain sufficient muscle mass for functional activities. But again, pleiotropy. Myostatin doesn't just affect skeletal muscle. It influences cardiac muscle, affects metabolism, has interactions with bone development. Turning it off is not a simple upgrade. It's a systemic biological change with consequences throughout the body. There's one more area of gene modification that I want to discuss in some detail, because it represents perhaps the most audacious and most philosophically challenging proposal in the entire genetic engineering for Mars conversation. And that is the question of engineering humans who can synthesize their own vitamin D without sunlight, and who can photosynthesize, even partially. Vitamin D is synthesized in human skin through a photochemical reaction triggered by ultraviolet B radiation from the sun. On Mars, the UV environment is actually more intense than on Earth in some wavelengths because there's less atmosphere to absorb it, but the intensity of usable sunlight for photosynthesis and vitamin D synthesis is lower because Mars is further from the sun. More importantly, Mars colonists spending most of their time indoors in pressurized habitats would get very little sunlight exposure. Vitamin D deficiency is already one of the health concerns for ISS astronauts. For long-term Mars colonists, supplementation would be necessary, but supplementation requires supply chains that may not always be reliable. Engineering humans who could synthesize vitamin D more efficiently, or who had reduced vitamin D requirements, would be a practical adaptation. But photosynthesis, that's where things get genuinely wild. There are a handful of animal species that harbor photosynthetic symbionts or that have actually incorporated photosynthetic genes into their own genome. The eastern emerald alicia, a sea slug, is one of the most famous examples. This slug can incorporate chloroplasts, the photosynthetic organelles of algae, into its own cells and survive for months by photosynthesizing, essentially running on sunlight. This process, called kleptoplasty, is not true genetic photosynthesis in the slug's own cells. It's borrowing organelles from eaten algae, but it points to the possibility that animal cells can at least maintain photosynthetic activity. More intriguingly, research has found genuine horizontal gene transfer of photosynthesis-related genes from algae to the slug genome, though the full functional significance of this is still debated. Some researchers have proposed, very speculatively, that engineering genes for photosynthesis into human cells could theoretically provide a supplementary energy source for Mars colonists, reducing their caloric requirements. This would be enormously difficult, possibly impossible with current or even near-future technology, because photosynthesis is a tremendously complex process involving hundreds of proteins working in concert in specialized organelles. But the theoretical interest is real, and some preliminary work on expressing plant photosynthesis proteins in mammalian cells has been attempted. The University of Wisconsin's John Bott, this is speculative research territory, and others have explored whether specific components of the photosynthesis pathway could function in mammalian cells. The results are early and modest, but the direction of inquiry is real. The idea of a partially photosynthetic human is one of those concepts that sits precisely at the boundary between science and science fiction, the boundary that keeps moving forward as technology advances. 20 years ago, CRISPR was science fiction. 10 years ago, a functioning CRISPR edit in a human embryo was science fiction. 5 years ago, the first CRISPR-based gene therapy approved for clinical use in the United States was science fiction. The boundary moves. It always moves. Now let's pivot to something that is perhaps even more immediately actionable than genetic engineering, but which sits in a fascinating relationship with it: epigenetics. Because one of the most important discoveries in biology over the past 30 years is that the genome is not destiny. The genome is a library. Which books you read, which genes you express, depends on epigenetic factors, chemical modifications to the DNA and to the histone proteins that DNA is wrapped around, that determine which genes are accessible and which are silenced. And crucially, epigenetic patterns can be modified by environment and experience, sometimes within a single lifetime, and sometimes they can be inherited across generations. The field of epigenetics has already revealed that the space environment modifies human epigenetics. The twin study showed that Scott Kelly's epigenetic patterns changed significantly during his year in space, with thousands of epigenetic marks shifting in ways that altered gene expression. Some of these changes involved genes related to the immune system, cognition, and aging. And here's the fascinating thing. Some researchers believe that targeted epigenetic modification, changing which genes are expressed in which cells without changing the DNA sequence itself, might offer a more reversible and therefore more ethically manageable approach to preparing humans for space than germline genetic modification. Epigenetic modification wouldn't change the genes passed to future generations, unless epigenetic inheritance is occurring, which is itself a frontier of research. It would change only the pattern of gene expression in the individual. An astronaut or colonist could have their epigenome specifically tuned for Mars before departure, enhancing the expression of relevant protective genes, dampening the expression of genes whose activity is counterproductive in the Mars environment. When and if they return to Earth, the epigenetic changes might partially or fully reverse. This is a much less permanent intervention than germline editing, and while it raises its own scientific and ethical questions, it sits closer to the realm of accepted medical practice than the more radical proposals. However, we know from research on transgenerational epigenetic inheritance that some epigenetic modifications can be passed to children and grandchildren through mechanisms that bypass the genetic code itself. Research in animal models has shown that stress, diet, and chemical exposures can leave epigenetic marks that persist for multiple generations. If Mars colonists' epigenomes are significantly modified by the Martian environment, those modifications might be partially inherited by their children born on Mars, even without any deliberate genetic engineering. The next generation of Martians might be biologically different from Earth humans, not just because of the direct effects of their own environment, but because of epigenetic inheritance from their Mars-adapted parents. This is a theory at this stage, and I want to be clear that we are in speculative territory here. Transgenerational epigenetic inheritance in humans remains a topic of active scientific debate, and the extent to which specific epigenetic modifications would be heritable is unknown. But it's a possibility that researchers take seriously, and it adds yet another layer of complexity to the question of how Mars colonists might diverge from Earth humans over generations. Now let's talk about something that rarely gets enough attention in these discussions, and that is the psychological dimension, because the mind is as much a biological system as the body, and the Mars environment would be profoundly, relentlessly challenging to human psychology. Consider what the psychological environment of a Mars colony would actually be like. You are 140 million miles from home on average, further at some orbital positions. Communication with Earth takes between 3 and 22 minutes each way, depending on where the planets are in their orbits, meaning a real-time conversation is impossible. You are in a small, enclosed habitat with the same small group of people for years at a time. You cannot go outside without wearing a suit. The landscape outside, while genuinely beautiful, is utterly alien, with no trees, no animals, no rain, no ocean, no other life of any kind visible to the naked eye. You cannot easily leave if things go wrong. You cannot call for help that will arrive quickly. You are, in the most profound sense of the word, isolated. The psychological effects of prolonged isolation, confinement, and monotony are well documented from Earth analogs, including Antarctic research stations, submarine deployments, and simulated Mars missions like the HIC's program in Hawaii, which conducted several isolation experiments of various durations. Research from these analogs shows consistent patterns: increased interpersonal conflict, decreased motivation, impaired cognitive performance, disrupted sleep, and in some cases depression and anxiety that are resistant to treatment. On a real Mars mission, these effects would be compounded by genuine mortal danger, the impossibility of rescue or quick return, and the knowledge that every system's failure is potentially lethal. Could genetics help here? The question sounds almost absurd, but some researchers are exploring it seriously. There is substantial evidence that genetic factors influence psychological resilience, stress tolerance, emotional regulation, and susceptibility to depression and anxiety. The serotonin transporter gene, the BDNF gene, which codes for brain-derived neurotrophic factor and plays a role in learning, memory, and mood, and the COMT gene, which regulates the clearance of dopamine in the prefrontal cortex and influences stress response, have all been studied in the context of psychological resilience. The evidence is complex, and the effect sizes of individual gene variants are generally small. But the principle that genetic factors contribute to psychological resilience is solid. More intriguingly, some researchers have speculated about engineering enhanced serotonin system function, or enhanced endocannabinoid system activity, or other neurochemical modifications that might make humans more psychologically resilient to the conditions of Mars. The idea of engineering emotional stability or psychological resilience into Mars colonists sounds immediately like something from a dystopian novel. And the ethical concerns are significant. Who defines what level of psychological resilience is desirable? Who controls which emotional responses are engineered? Is engineering emotional regulation in human beings compatible with human autonomy and dignity? These concerns are not hypothetical. History has too many examples of systems that tried to engineer psychological compliance in human populations, usually through coercive means rather than genetic ones, but the moral structure of the concern is similar. Any proposal to engineer the psychological characteristics of human beings, even with the best intentions, needs to be approached with extraordinary caution and subjected to the most rigorous ethical scrutiny. Yet the psychological challenge of Mars is real, and it cannot be solved by better habitat design alone. The human mind was built for a world with seasons and rain and other species and long-term social relationships with large communities, none of which Mars currently offers. Some kind of adaptation, whether through training, pharmacology, therapy, or eventually genetics, will be necessary. Let's change gears now and talk about something fascinating and slightly unexpected: the actual microbes. Not the gut microbiome this time, but the possibility that Mars might itself harbor life, and what that would mean for the genetic engineering conversation. The question of whether Mars currently harbors life, or whether it harbored life in the past, is one of the most important and most carefully avoided conversations in mainstream space exploration. The scientific evidence for past habitability of Mars is now overwhelming. We know that Mars had liquid water on its surface for hundreds of millions of years, possibly over 1 billion years, early in its history. We know that it had a thicker atmosphere and a magnetic field that protected it from radiation. We know that it had a warmer climate. The conditions for life as we know it, liquid water, energy sources, the necessary chemical elements, were all present on early Mars. Whether life actually arose under those conditions is the question we don't have an answer to. But there are some intriguing hints, none of them definitive. The Curiosity rover detected methane in the Martian atmosphere in 2014, a finding that was both exciting and puzzling. Methane shouldn't be there. Ultraviolet radiation from the sun destroys methane in the Martian atmosphere very quickly, so any methane present must be continuously replenished. The most exciting possible source would be methane-producing microorganisms, methanogens, living in the subsurface. But methane can also be produced by geological processes, so this observation is far from conclusive. The methane findings have been controversial. The ExoMars Trace Gas Orbiter, launched by the European Space Agency and specifically designed to study trace gases in the Martian atmosphere, initially reported no detectable methane in 2019, creating a puzzling contradiction with the Curiosity measurements. The debate continues. In 2020, researchers using radar data from the European Space Agency's Mars Express orbiter published a paper in Nature Astronomy reporting evidence for a liquid water lake beneath the south polar ice cap of Mars, at a depth of about 1.5 kilometers, kept liquid by the salt content of the ice above and possibly by geothermal heat from below. The existence of a subsurface liquid water environment, even a briny, frigid one, immediately raised the possibility of microbial life. The discovery was exciting, though subsequent analyses have suggested that the radar data might also be explained by other geological features, and further verification is needed. Why does this matter for the genetic engineering conversation? Because if Mars has life, even microbial life, the ethics and the biology of human modification for Mars get dramatically more complicated. If Mars has its own biosphere, even a microscopic one, then sending genetically modified humans there introduces a new set of questions. Could engineered human organisms, with their modified genes and engineered microbiomes, contaminate Martian life and destroy our chance to study it? Could Martian microorganisms interact with genetically modified human biology in ways that were impossible to predict? The planetary protection protocols that govern space missions, designed to prevent biological contamination of other worlds and to prevent the inadvertent return of alien life to Earth, would need to be completely rethought for a scenario involving genetically modified colonists. Some biologists have proposed something even more provocative. What if the genetic engineering went in the other direction? What if, after finding that Mars harbors microbial life with unique biochemical capabilities, we studied those Martian organisms and then used genetic engineering to introduce their most useful traits into human biology? What if Martian life had evolved radiation resistance mechanisms, or cold tolerance mechanisms, or biochemical pathways that humans could use? The idea of incorporating Martian genetic material into human biology sounds absolutely extraordinary, but it follows the same logic as the tardigrade and Deinococcus examples we discussed earlier. If a gene works, if a biological trick is useful, it doesn't matter which organism originally evolved it. Of course, this assumes that Martian life uses the same genetic code as Earth life, which is not guaranteed. Most astrobiologists believe that if life exists on Mars, it could have originated from the same source as Earth life, since we know that rocks and material regularly exchange between the two planets through meteor impacts, a process called panspermia. If Martian and Earth life share a common ancestor, they might use the same basic genetic machinery, DNA and RNA and proteins with similar structures. In that case, horizontal gene transfer between Martian and Earth organisms, or the engineering of Martian genes into human cells, would at least be feasible in principle. But if Martian life evolved independently, if it uses a different genetic code, different biochemistry entirely, the integration would be orders of magnitude more complex. This thought experiment, the discovery of Martian life as a new source of genetic tools for human Mars adaptation, illustrates something important about the whole conversation we're having. Genetic engineering for Mars is not a closed-ended project. It's not like building a rocket where you have a fixed endpoint. It's an open-ended exploration of biological possibility, one that would interact in unpredictable ways with every other piece of knowledge we gain about Mars, about extremophiles, about the limits of life itself. Now let's talk about something that has been implicit throughout this entire discussion, but that deserves explicit attention: the economics and the politics. Because no technology exists in a vacuum, and the question of who controls genetic engineering for space, who makes the decisions about which modifications are made, who has access to the technology, and who bears the risks, is as important as any of the biology. The private space industry is, at this point in history, driving Mars ambitions more than any government agency. SpaceX, Blue Origin, and several other companies are working towards cislunar and eventually interplanetary transport. China's space agency, CNSA, has also made Mars a priority, having successfully landed the Zhurong rover in 2021. The landscape of space exploration is becoming more complex, more competitive, and more commercially driven than at any previous point in history. In this context, who would control the decision to genetically engineer Mars colonists? Would it be governments? Would it be the private companies that own the spacecraft and the habitats? Would it be the colonists themselves? And here's a disturbing possibility. If a private company is paying for your transport to Mars and your habitat and your food and your life support systems, do they have leverage over your biological choices? Could a future Mars colonization company offer genetically modified colonists better survival odds, and therefore implicitly pressure all colonists to accept modification? Could there be a two-tier system where modified colonists have access to certain opportunities and unmodified ones don't? These scenarios are not as far-fetched as they might sound. Employer influence over employee health decisions is already a contentious issue on Earth. The power dynamic between a Mars colonization company and its colonists would be so extreme as to make normal employment relationships look positively egalitarian. A colonist who refuses a modification that the company considers essential for mission success can't simply quit and go home. They can't look for another job. Their very survival depends on the company's infrastructure and goodwill. The potential for coercion, even subtle coercion, is enormous. The bioethics community has been raising these concerns for years. Françoise Baylis, a philosopher and bioethicist at Dalhousie University who has written extensively on genome editing ethics, has emphasized that any framework for human genetic modification must be grounded in genuine autonomy. Not the theoretical autonomy of someone who has signed a consent form, but the real autonomy of someone who has genuinely free choices. In a Mars colonization context, genuine autonomy might be essentially impossible to achieve. Conversely, some argue that the colonists themselves, having volunteered for the mission with full knowledge of its risks, have the right to choose genetic modifications for themselves and to make reasonable decisions on behalf of children they choose to have on Mars. The principle of reproductive autonomy, the right of individuals to make their own reproductive choices, is a cornerstone of medical ethics. If a couple wants to genetically modify their embryo to give it the best chance of surviving in the environment where it will be born and raised, are they not exercising reproductive autonomy? Is that so different from a couple choosing a prenatal diagnosis and making decisions based on the results? The answer depends on where you draw the line between accepted medical decision-making and novel territory that requires broader societal consent. And that line is not fixed. It shifts with technology, with culture, with scientific understanding, and with the accumulating experience of consequences, good and bad. There's a moment I want to take here to discuss what might be the most emotionally powerful and philosophically resonant aspect of all of this. What it means to be the first humans to be born on Mars. The first generation of children who open their eyes on a world that is not Earth, who grow up knowing no other sky, no other gravity, no other air, who look up through the transparent dome of their habitat at a star-filled sky where one of the brightest objects is a blue-green, and are told that that's where their species came from. Those children, whether or not they are genetically modified, will be different. The environment alone will shape them in ways we can't fully predict. If they have never experienced Earth's gravity, their bodies will be adapted to Mars. If they have never breathed unprocessed air, their immune systems will be calibrated differently. If they have grown up in an enclosed community of a few thousand people, their psychology will be shaped by that intimacy. If they have grown up knowing that their world requires constant technological intervention just to be habitable, their relationship to technology will be fundamentally different from any human being who has ever lived. And if they have been genetically modified, if their DNA was deliberately altered before they were born to give them better odds of surviving and thriving in their world, they will know that. They will know that they are, in a very specific and intentional way, different from the baseline human. How will they feel about that? Will they feel like failures of humanity, like deviation from the template? Or will they feel like the fulfillment of human potential, the next step in a story of adaptation that has been going on for 4 billion years? Research on how humans adapt psychologically to knowledge of their genetic status, from studies of people who have discovered they carry the BRCA gene for breast cancer to people born with Down syndrome, suggests that the response is extraordinarily variable and almost always more nuanced than outsiders expect. Identity is remarkably resilient and adaptable. People build meaning around who they are, whatever that is, with remarkable creativity. The first genetically modified Martians would likely be no different in that fundamental respect. They would find ways to be proud of who they are, even if who they are was partly design. In fact, there's a possibility that feels almost mythological in its grandeur, that the first Martian-born, Mars-adapted humans might look back at their genetic modification not with resentment but with gratitude. They might see it the way we might imagine a future generation looking at the vaccines that protected their ancestors from smallpox and polio, as a gift, as evidence that their predecessors cared about them enough to do something difficult and controversial and unprecedented so that they could live. Or they might not. They might look at it with anger, with a sense of violation, with the feeling that something was taken from them before they were born, that the pristine human template that was their birthright was altered without their consent. That is also a very human response, and it deserves to be taken seriously. Let's now address a question that is often raised in these discussions, and that I think requires more careful treatment than it usually receives. Couldn't we just build better technology instead? Couldn't we build habitats that protect colonists from radiation, suits that maintain perfect Earth-like conditions, exercise machines that prevent bone and muscle loss, and supply chains that deliver everything humans need, rather than modifying humans themselves? The answer is yes, to some extent. This is in fact the current plan for Mars. Any near-term Mars mission would rely entirely on technology for life support. And technology has advantages that genetic engineering currently lacks. It's testable, adjustable, reversible, and doesn't require human subjects. If a habitat design fails, you can fix it. If a radiation shielding material doesn't work well enough, you can try another one. If an exercise protocol is insufficient to prevent bone loss, you can modify the protocol. None of that requires touching anyone's genome. But technology has its own profound limitations in the Mars context, and understanding them is essential to understanding why the genetic engineering conversation is actually happening. First, mass and energy constraints. Getting anything to Mars is extraordinarily expensive in terms of mass and therefore energy. Every kilogram you send to Mars costs roughly tens of thousands of dollars at current launch costs, and even with the optimistic projections for SpaceX's Starship, it will be far from cheap. The more you rely on Earth supply technology, the more mass you need to ship to Mars, and the more vulnerable you are to the failure of that supply chain. A self-sustaining Mars colony that relies on imported life support technology is not truly self-sustaining. It's dependent. Second, the time horizon. If the goal is a Mars colony that can survive on its own for centuries or millennia, the idea of relying on technological solutions that must be continually manufactured, maintained, upgraded, and replaced with Earth-derived materials is fundamentally unstable. Technology fails. Complex systems fail. The history of technology on Earth is a history of constant maintenance and constant failure, usually with readily available replacement parts and expert repair personnel. On Mars, 100 million miles from the nearest hardware store, the failure of a critical life support system is an extinction event for the colony. Biological solutions, if they work, are more robust because they are self-replicating. A genetically modified human who passes their modifications to their children is a solution that perpetuates itself. Third, quality of life. Even if all the technological solutions work perfectly, the life of a Mars colonist relying on them is extraordinarily constrained. You can never go outside without a suit. You can never eat food that isn't carefully processed and tracked. You can never breathe air that doesn't come from a machine. You live your entire life inside. That's not just uncomfortable. It's psychologically damaging. It's not the kind of life that sustains a civilization. For Mars to be a true second home for humanity, for people to live there not just as a scientific outpost, but as a home they love and want to be in, the environment needs to be more livable. And the only way to make the environment of Mars livable without technological mediation is either to terraform it, which is a multi-thousand-year project even in optimistic scenarios, or to adapt the human inhabitants to it. The terraforming option, transforming Mars into a more Earth-like planet through planetary engineering, is itself a subject of intense scientific debate. The basic idea is to increase Mars's atmospheric pressure and temperature, to melt the polar ice caps and release CO2 and water vapor, to establish a greenhouse effect that warms the planet, and eventually to introduce oxygen-producing organisms to create a breathable atmosphere. Proposals have ranged from manufacturing and releasing trillions of tons of super greenhouse gases into the Martian atmosphere to positioning giant space mirrors to focus additional sunlight on the poles. Stephen Hawking was a proponent of Mars terraforming, as is Elon Musk, who has famously suggested detonating nuclear bombs at the Martian poles to release CO2 from the ice caps, a suggestion that most planetary scientists consider both impractical and potentially counterproductive. The problem with terraforming is time. Even the most optimistic models suggest that creating a breathable, warm Mars with liquid surface water would take at least several hundred years, possibly thousands, even with aggressive technological intervention. And more recent research suggests that it might be fundamentally impossible to terraform Mars to Earth-like conditions with any currently conceivable technology, because Mars doesn't have enough CO2 in its reservoirs to create a thick enough greenhouse atmosphere even if all of it were released. A study published in Nature Astronomy in 2018 by Bruce Jakosky of the University of Colorado and Christopher Edwards found that even releasing all the CO2 available from all sources on Mars, including polar ice and subsurface reserves, would only increase atmospheric pressure to about 1.2% of Earth's, far too thin to be breathable and inadequate to maintain liquid water on the surface. So terraforming, at least to the full Earth-like standard, is either centuries away or physically impossible with current knowledge. This is why some researchers have coined the term paraforming as an alternative concept. Instead of transforming Mars to suit humans, transform humans to suit Mars. Paraforming Mars means adapting the inhabitants, not the planet. And genetically engineering those inhabitants is the most extreme and most capable version of paraforming. The word paraforming is used in some research contexts and has been discussed by researchers including Robert Freitas, the author of the 4-volume Nanomedicine series, and by thinkers in the transhumanist tradition, though it hasn't yet entered mainstream scientific discourse. But the concept captures something important. The assumption that terraforming is the only approach to making Mars habitable is just that, an assumption. It's not an inevitable truth. The history of life on Earth is a history of organisms adapting to environments, not environments adapting to organisms. Now let's talk about a specific research program that represents perhaps the most serious and systematic current approach to the question of genetic engineering for space. The work being done at the Center for the Utilization of Biological Engineering in Space, known as CUBES, a consortium of researchers funded by NASA and based at the University of California, Berkeley. CUBES is focused on developing biotechnology solutions for long-duration space missions, including the development of biological systems for life support, food production, pharmaceutical synthesis, and material production in space. While the center's current work is focused more on engineering microorganisms and plants for space than on engineering humans, it represents the integration of cutting-edge biotechnology into the serious planning for space exploration. The approach at CUBES and similar programs illustrates an important point about the genetic engineering for Mars conversation. It's not binary. It's not a choice between sending unmodified humans to Mars in suits versus sending fully genetically redesigned Martian superhumans. There's a spectrum of biological intervention that ranges from engineering the microorganisms used in life support systems, to engineering the food crops grown on Mars, to engineering the human gut microbiome for Mars conditions, to engineering specific protective traits into the human genome itself, to full-scale germline modification of Mars colonist lineages. Each step on this spectrum raises additional technical challenges and ethical questions. But each step is also more achievable, more incrementally justifiable than the full endpoint. We might begin by engineering the biological systems that support human life on Mars, rather than the humans themselves. A Mars habitat's life support could be largely biological rather than purely mechanical, with engineered microorganisms recycling waste, producing oxygen, synthesizing vitamins, maintaining atmospheric composition, and even generating electricity through biologically mediated processes. The plants grown in Martian greenhouses might be engineered for ultra-high nutritional yield, radiation resistance, low water requirements, and the ability to grow in Martian regolith with minimal Earth-derived supplements. These biological systems would form a kind of engineered biosphere around the human colonists, a Mars-adapted life support ecosystem, even while the humans themselves remained relatively unmodified. This approach is currently the most scientifically mature part of the biological engineering for Mars conversation. Researchers have already begun developing engineered organisms for space applications. In 2020, a study published in Nature Communications showed that bacteria engineered to produce essential amino acids and vitamins could potentially serve as a nutritional supplement for Mars colonists, reducing the volume and mass of food supplements that would need to be imported from Earth. In 2019, researchers from the German Aerospace Center demonstrated that several species of bacteria and archaea could survive in simulated Martian conditions, including low pressure, low temperature, and high radiation, suggesting that engineered extremophilic microorganisms might be able to function in Mars's actual environment, not just in Earth laboratories simulating it. The distinction between engineering the environment and engineering the human is philosophically important. We generally feel more comfortable with the idea of modifying our tools and our environment than with modifying ourselves. We've been engineering our environment for 10,000 years of agriculture and 10,000 years before that of controlled fire use. Engineering biological life support systems for Mars is an extension of this tradition. Engineering the humans themselves is a different kind of step, and most people feel the difference even if they struggle to articulate exactly why. But let's push on that feeling for a moment, because it might be more arbitrary than it seems. Here's an argument. The human gut microbiome is already engineered, in a sense, by the foods we eat, the antibiotics we take, the environments we live in. When we deliberately select which microorganisms colonize our gut by eating certain foods or taking probiotics, we are engineering our internal biology. The fact that this happens at the microbial level rather than the genomic level doesn't make it categorically different. And if we deliberately engineer an entire custom gut microbiome for Mars colonists, the line between that and engineering the colonist's own genome gets blurrier. Where exactly does the self end and the environment begin? Is the gut microbiome part of you or part of your environment? By most biological measures, it's part of you. But it can be modified externally. The boundaries are already fuzzy, and they will only get fuzzier as biotechnology advances. Let's talk for a moment about the timeline. Because one of the most important questions in this whole discussion is whether any of this is actually going to matter in the next relevant time frame, meaning the next 20 to 50 years when Mars missions and potentially early colonization might actually occur. The answer is nuanced. The genetic engineering for space conversation operates on multiple timescales simultaneously. In the short term, the next 10 to 20 years, the most relevant biotechnology applications for Mars will not be human genetic modification. They will be the development of engineered organisms for life support, the improvement of CRISPR-based gene therapies for treating space-related illnesses, and the development of pharmacological interventions to counteract the physiological effects of microgravity and radiation. These are all achievable within the current state of technology and ethical frameworks. In the medium term, 20 to 50 years, if Mars colonization is actually underway with long-duration missions, the biological challenges will be urgent enough that more aggressive interventions will be considered. This is the time frame in which epigenetic modification, engineered gut microbiomes for Mars, and possibly somatic, non-heritable, genetic modifications to individual astronauts might be deployed, particularly if the evidence of irreversible damage to unmodified colonists makes the case for intervention compelling. The ethical debates will be fiercer, the science more mature, and the public more engaged. In the long term, 50 to 100 years and beyond, if Mars has established colonies and those colonies are growing, the question of germline modification for future generations will be unavoidable. Not necessarily because anyone wants to modify human germlines, but because the forces of natural selection will be operating on the Martian population, and every generation of Mars-born humans will be slightly different from their Earth-born ancestors based on who survives and reproduces successfully in the Martian environment. Whether or not we deliberately modify anyone, evolution will be doing its own slow, imprecise version of genetic engineering. The question will be whether to guide that process deliberately or let it proceed randomly. Stephen Petranek, whose book How We'll Live on Mars influenced the Netflix documentary series Mars, has argued that within 100 years, Mars colonists will be biologically distinguishable from Earth humans, primarily through the accumulated effects of adaptation to lower gravity, higher radiation, and different diet. Whether or not genetic engineering is deliberately employed, the divergence will happen. The question is whether it happens in a way that maximizes health and well-being for the colonists, which would benefit from deliberate guidance, or in a way that is dictated purely by which random genetic variations happen to confer survival advantages in the Martian environment, which is crueler and less predictable. Natural selection is not kind. It doesn't care about individual well-being. It cares only about differential reproductive success. The genetic variants that get passed to future generations on Mars are not necessarily the ones that lead to the healthiest, happiest lives. They're the ones that lead to the most offspring. Those could be the same thing, or they could be very different things. In the history of life on Earth, they have often been very different things. Evolution has produced organisms optimized for reproduction in their environment, but also organisms that suffered terribly from the byproducts of that optimization. Natural selection, left to its own devices on Mars, might produce future Martians who are reproductively successful in the Martian environment, but who experience vastly increased rates of cancer from radiation damage, who age prematurely, who develop bone and muscle disorders in middle age, and who die younger than their Earth counterparts. The alternative to deliberate genetic guidance is not the pristine, unmodified human ideal. It's random evolutionary selection with no conscience. There's one more thing I want to address before we start to draw the story toward its conclusion, and that's the question of what the scientific consensus actually is, right now, today, on the feasibility of genetic engineering for Mars adaptation. The honest answer is that it is a topic of serious scientific discussion, but one where the science is very early, the ethical issues are profound, and there is no consensus on a path forward. The scientific community is broadly in agreement on several points. First, the biological challenges of Mars colonization are severe and cannot be solved by technology alone in the long run. Second, genetic engineering technology, particularly CRISPR-based systems, is advancing rapidly and will likely become powerful enough to attempt complex human modifications within the next few decades. Third, there are real biological mechanisms, drawn from extremophile organisms and natural human genetic variation, that could potentially be engineered to improve human survival in the Mars environment. Fourth, the risks of current genetic engineering technology for complex organisms are still too high and the off-target effects too unpredictable for human applications beyond treatment of serious diseases. The disagreements are on the ethical questions, the timeline, and the priorities. Some researchers, like Christopher Mason, argue that we should be developing the genetic toolkit for Mars now, preparing the scientific groundwork so that if we decide to use it, the tool is ready. Others argue that we should focus all our biotechnology effort on making the non-human biology of Mars colonization work, the life support systems, the food production, the pharmaceutical manufacturing, and leave human modification as a last resort. Still others argue that any human genetic modification, beyond treatment of specific diseases, is ethically impermissible regardless of context, and that the Mars challenge should be met with better technology, not modified humans. There is also a growing voice from the disability rights community that deserves recognition in this conversation. The disability rights movement has long argued against the assumption that any physical difference from the standard human template is a problem to be fixed, and that engineering future humans to meet some ideal physical standard is itself a form of discrimination against those who don't meet that standard. The application of this perspective to genetic engineering for Mars is complex, because the modifications being discussed are not about eliminating disability but about enabling survival. But the underlying concern that genetic modification projects embed assumptions about what kinds of human bodies and minds are acceptable is a valid one that the mainstream discussion sometimes sidesteps. Indigenous scholars and ethicists have also raised important questions about the cultural dimensions of Mars colonization. The framing of Mars as empty territory waiting to be colonized by humanity recapitulates historical colonial narratives in ways that some scholars find deeply troubling. If that broader colonial framing is already problematic, what does it add to argue that we should modify the humans doing the colonizing to be better suited to the territory? The colonial metaphor is not perfect. Mars is genuinely uninhabited as far as we know, but the power structures and assumptions embedded in colonization narratives are worth examining even when the territory in question has no indigenous inhabitants. Now, one of the most thought-provoking things about all of this, the thing I want to leave you with as we move toward the end of the story, is the recognition that we are standing at a genuinely unique moment in the history of life on this planet. In 4 billion years of evolution, there has never been a species capable of deliberately reshaping its own genome to prepare for life in a different world. We are the first. We are the only species, in all the billions of species that have ever existed on Earth, with the cognitive and technological capacity to look at our own biology, understand it mechanically, and consider rewriting parts of it on purpose. That is extraordinary. It is humbling and terrifying and magnificent all at once. We did not ask to be in this position. We arrived here through the accumulation of scientific discoveries, technological developments, and cultural choices stretching back centuries. But here we are. And the decisions we make in the next few decades about whether and how to use the power of genetic engineering in the context of space exploration will echo for thousands of years. Consider the Voyager Golden Record, that copper disk that NASA attached to the Voyager probes in 1977, containing sounds and images of life on Earth, intended as a message to any alien civilization that might one day find it. On that record are greetings in 55 languages, music from Bach and Chuck Berry and Indian ragas and Peruvian pipes, images of humans and animals and cities, the sound of a mother's first words to her newborn child. It was a message in a bottle thrown into the cosmic ocean, a statement of what humanity is at this moment in its history. Now imagine that our genetic engineering of Mars colonists is a different kind of statement, one written not in copper and gold, but in adenine and guanine and cytosine and thymine. A statement that says, we were here on Earth, but we chose to go there, and we changed ourselves to be able to do it. What do we want that statement to say? What kind of beings do we want to send into the cosmos? What kind of beings do we want to become? These are questions that don't have scientific answers. They have human answers, and arriving at them requires exactly the kind of broad, inclusive, emotionally engaged public conversation that this story is trying to contribute to. The scientists and the bioethicists and the policymakers cannot make these decisions alone. They are decisions about human identity, about the meaning of our species, about what we owe future generations and what we have the right to choose for them. They are your decisions, too. Something I find particularly beautiful and haunting in all of this is the possibility that the moment we send the first genetically modified humans to Mars, we will have created the first members of what might eventually become a new human lineage, people as far from us, in biological terms, as we might someday be from them. And there's a good chance that those beings, those future Martians, will never set foot on Earth, will never feel its gravitational pull, will never stand in its rain, will never breathe its air without technological assistance. The planet that built us, that spent 4 billion years producing us, will be to them what the African savanna is to a person who has never left Tokyo. The ancestral homeland, known through stories and images and genetic memory, but never directly experienced. Is that loss? Is that tragedy? Or is that simply the continuation of the most human thing there is? The endless, restless, irresistible urge to go somewhere new and build something there. Our ancestors left Africa. They crossed the Sahara and walked to the Middle East and kept going, across Asia, across Europe, over the Bering Land Bridge into the Americas, and eventually onto every island that human curiosity and necessity could reach. They didn't have genetic modifications. They had curiosity, determination, and the deep human conviction that whatever is over the next horizon is worth knowing about. The Mars pioneers who are alive right now, who are building the technology and writing the scientific papers and having the ethical debates, they are the descendants of those earliest wanderers. They carry in their DNA the record of every migration and every adaptation and every improbable survival their ancestors achieved. And now we are looking at Mars, that pale red in the night sky that humans have watched for thousands of years and given names to in dozens of languages, that the ancient Babylonians named for their god of war, and the Egyptians called Herdesher, and the Persians called Bahram, and we call by the name of the Roman deity of conflict and force. And some of us are asking, what do we need to become to go there? What is it worth to be something new in a new world? Let me take you somewhere specific now, a place where the philosophical speculation meets the laboratory bench, because this story has been asking big questions, and it's important to remember that those big questions are also being addressed in very specific, concrete, technical ways by real people in real laboratories right now. At the Weiss Institute for Biologically Inspired Engineering at Harvard University, researchers are working on what they call genetic circuit design, the ability to engineer genetic sequences that function like electronic circuits, turning on in response to specific signals and turning off again when those signals disappear. One potential application of this technology is radiation-responsive genetic circuits that could activate protective genes specifically in response to radiation exposure. Instead of having enhanced radiation protection genes active all the time, which might have metabolic costs or side effects, you would engineer cells to turn on their radiation defenses only when needed and turn them off again when the radiation exposure drops. This is an elegant solution that draws on principles from electronic engineering and applies them to biology, and it illustrates the sophistication of the current state-of-the-art in genetic circuit design. At MIT's Media Lab, researchers associated with the Space Exploration Initiative have been exploring a range of technologies for human enhancement for space, ranging from wearable technology to pharmacological approaches to, yes, genetic ones. The MIT Space Exploration Initiative is notable for its interdisciplinary approach, bringing together engineers, biologists, designers, artists, and social scientists to think about what human beings will need, physically and psychologically, in space environments. At UC San Francisco, one of the world's leading centers for CRISPR research, researchers have been developing more precise and efficient versions of CRISPR editing tools, including base editors that can change individual DNA letters without making double-strand cuts, which reduces the risk of off-target effects, and prime editors that can insert new genetic sequences with even greater precision. Each advance in CRISPR precision reduces the risk of unintended consequences, making more complex genetic modifications more feasible and more safe. At Stanford's Department of Genetics, researchers are working on understanding the genetic basis of human longevity and age-related disease, knowledge that is directly relevant to Mars colonization. The same genetic factors that determine how gracefully humans age on Earth would also influence how they cope with the accumulated damage of radiation, low gravity, and other Mars-specific stressors over a lifetime. Understanding the genetics of resilience and aging could point towards specific interventions that enhance colonist health over decades. And in Beijing, at the Beijing Genomics Institute, which is one of the world's largest genomics research organizations, work is ongoing on large-scale genomic analysis of extreme environments and the organisms that live in them, including Tibetan highlanders, Antarctic microorganisms, and deep-sea extremophiles. The database of genetic adaptations to extreme environments being assembled by institutions like BGI will be an essential resource for any future program of genetic engineering for space, providing the biological vocabulary from which a Mars adaptation toolkit might be assembled. The global nature of this research is itself significant. The conversation about genetic engineering for Mars is not just a Western conversation, not just an American one. It's happening in China, in Europe, in Russia, in Japan, in Australia, in India. Different cultures bring different values to the ethical questions, different assumptions about individual rights versus collective goals, different ideas about what counts as acceptable modification of human nature. The He Jiankui case was striking in part because of the specific cultural and regulatory context in which it occurred, where the pressure to achieve scientific priority, combined with regulatory gaps and cultural attitudes toward genetic intervention, created a space in which something happened that shocked the global scientific community. The international nature of the research means that international governance frameworks are essential, but they are also extraordinarily difficult to achieve, as we have seen with every other attempt to create global governance of transformative technologies. The 2015 International Summit on Human Gene Editing, organized by the National Academies of Sciences in the United States, the Royal Society in the UK, and the Chinese Academy of Sciences, produced a statement calling for a moratorium on clinical germline editing until safety, efficacy, and societal consensus could be established. A subsequent summit in 2018, convened just weeks after the He Jiankui announcement, produced a stronger statement condemning what he had done and calling for an international framework. A third summit in 2023 continued this conversation, with a growing consensus that the scientific community needs to develop specific standards for evaluating proposals for germline editing, even while the moratorium on clinical applications remains in place. These summits represent the beginning of a global governance conversation, but only the beginning. The pace of technological development is fast, the commercial incentives are significant, and the regulatory landscape is fragmented across national jurisdictions. The history of dual-use technologies, from nuclear power to the internet to artificial intelligence, suggests that governance frameworks always lag behind technical capabilities, and that the consequences of that lag can be severe. For genetic engineering for space, the lag might not matter much in the next decade, because the technology is not yet at the point where human germline editing for space adaptation is clinically feasible or safe. But within 20 years, within 30, the gap between what can be done technically and what has been collectively decided to be permissible could become uncomfortably narrow. There's a science fiction framework that might be useful here, not because it's predictive, but because it captures the emotional and ethical stakes in a way that pure scientific discourse sometimes doesn't. The film Gattaca, released in 1997 and set in a near future where genetic engineering of embryos is routine, depicts a world divided between the valids, who were genetically optimized at conception, and the invalids, who were born naturally and are systematically disadvantaged in access to education, careers, and social status. The film's protagonist, born invalid, is determined to be selected for a mission to Saturn's moon Titan, something the system says he cannot achieve. The film is a meditation on the relationship between genetic determinism and human will, and it remains one of the most thoughtful explorations of the social consequences of genetic engineering ever produced. It's worth noting that Gattaca's world is not one of genetic engineering for survival in an alien environment. It's one of genetic enhancement for competitive advantage in a terrestrial world. The Mars adaptation context is meaningfully different in motivation, if not in the technical process. But Gattaca's warning is not primarily technical. It's social. It's about the creation of a caste system based on genetics, about the way in which good intentions in genetic selection can become the foundation of profound injustice. Any plan for genetic engineering in the context of Mars colonization needs to grapple seriously with that warning. Here's another thought experiment. Imagine it's 2150. There are 40,000 people living on Mars. They are 3 or 4 generations removed from Earth, and most of them have never been to Earth. Their families were among the first pioneers, and many of them carry the genetic modifications, whether deliberate or selected by natural circumstances, that allowed their ancestors to survive in the Martian environment. They are demonstrably healthier on Mars than an unmodified Earth human would be. They can tolerate higher radiation doses. Their bones maintain density without Earth-level gravity. Their gut microbiomes are engineered for Mars agriculture products. They are, in various medical and biological senses, better adapted to their world than any of us would be. Now imagine that a ship from Earth arrives with a new group of colonists, none of whom are genetically modified, who are there on a new colonization program. They need medical care almost immediately because Mars is harder on their bodies. They need more resources because their life support needs are greater. They are, in the Martian context, less capable than the native-born Martians. How does the native Martian community treat them? What power dynamics emerge? What does it mean, in that context, to be the unmodified human in a world where the modified ones have the home advantage? Flip it around. Back on Earth, a Martian-born colonist arrives for the first time. Maybe they're there for negotiations, or cultural exchange, or to meet family they've never seen. Earth's gravity is crushing them, 3 times heavier than they've ever experienced. Earth's atmosphere feels overwhelming in its richness. Earth's microbial environment, with which their immune system did not co-evolve, causes health problems. They are the alien now, not in terms of legal status, but in terms of biology. How does that feel? How does Earth receive them? These are not idle speculations. They are the lived futures that the decisions being made today in genetics laboratories and space policy offices will create. And they deserve to be imagined as vividly and as seriously as we imagine the engineering challenges. We are now at a point in this story where I want to bring together the threads and see what they form. Because this is an enormously complex topic, and I think it's worth pausing to take stock of what we know, what we don't know, and what it means. What we know is this. Mars is genuinely, profoundly hostile to human biology in ways that cannot be fully addressed by technology alone, at least not in the long-term colonization scenario. The 3 most critical biological challenges are radiation, gravity, and a hostile atmosphere, and all 3 are addressed at best incompletely by current and near-term projected technology. Genetic engineering technology, particularly CRISPR and its successors, is advancing rapidly and becoming more precise and more capable with each passing year. There are real biological mechanisms, drawn from extremophile organisms and natural human genetic variation, that could theoretically be engineered to improve human survival in the Mars environment. Experiments in cell culture and animal models have demonstrated that some of these mechanisms can be introduced into non-native biological systems and function there. The ethical questions surrounding any application of this technology to humans, particularly in the germline, are profound and cannot be resolved by science alone. What we don't know is also important. We don't know whether any of the specific modifications proposed for Mars adaptation would actually work safely in a complex human organism. We don't know whether the pleiotropic effects of the proposed modifications would be manageable or whether they would create new problems worse than those they solved. We don't know what long-term partial gravity does to the human body, because we've never had humans in that environment for extended periods. We don't know whether Martian life exists, and if it does, we don't know how it would interact with genetically modified human biology. We don't know whether the social and political frameworks necessary to govern genetic engineering for space can be developed in time to be relevant. What it means is the hardest part. It means that we are a species facing a challenge that our biology did not prepare us for, but that our intelligence, our curiosity, and our extraordinary capacity for both creation and destruction has made possible. Whether to use the power of genetic engineering to meet that challenge is not just a scientific question or a technical question. It is a question about values, about what we are willing to do to ourselves and to future generations, about where we draw lines between acceptable and unacceptable intervention in the human blueprint, about who gets to make those decisions and on whose behalf. It means that the conversation happening in genetics labs and bioethics journals and space agency meeting rooms is arguably the most important conversation of our era, and it's happening largely out of view of the public who will be most affected by its conclusions. And it means that stories like this one, attempts to bring this conversation into the open, to explain it in human terms, to make it emotionally real rather than abstractly scientific, matter more than ever. Because the decisions that will determine whether humanity's future includes a modified, adapted, Mars-surviving lineage will not be made only by scientists. They will be made by the cultures that scientists live in, the democratic systems that fund and regulate their work, the public conversations that shape what is considered acceptable and what is not. You, listening to this right now, are part of that conversation. What do you think? Do you believe that humans should genetically modify themselves for Mars? Do you think the survival of the species as a multiplanetary entity justifies the ethical risks of germline modification? Do you think we should focus all our resources on building better technology and leave human biology alone? Do you think the colonists themselves should have the right to choose modification for themselves and their children? These are not rhetorical questions. They are real ones, and the answers matter. Ask yourself, if you were offered the chance to be genetically modified for a Mars mission, if you knew that without modification you would experience significantly higher cancer risk, significant bone and muscle loss, and potential cognitive effects from radiation, but with modification those risks would be substantially reduced, would you say yes? And if you would say yes for yourself, does that mean you would say yes for a child you were planning to have on Mars? Let me tell you about one more piece of research that I find particularly moving as we move toward the end of this story. In 2021, researchers published a paper in Astrobiology examining the minimum viable population size for a self-sustaining Mars colony, taking into account both the demographic and genetic factors necessary for long-term survival. Their analysis suggested that a minimum viable population for Mars would be somewhere between 110 people on the very low end, under highly specific conditions, and more realistically around 500 to 2,500 people for a truly self-sustaining colony. The genetic factors considered include the minimum necessary genetic diversity to avoid the inbreeding depression and genetic drift that would otherwise doom a small founding population over centuries. This is population genetics, and it's been applied to island colonization on Earth and to the founding populations of other isolated communities. But it's now being applied to Mars, and the implications are both beautiful and sobering. The beauty is this: every Mars colony that survives and grows will carry within itself the genetic seeds of a new human lineage. The diversity of genes in those first few 100,000 pioneers will shape the entire genetic future of Martian humanity, perhaps for thousands of years. The founders of a Mars colony will be, in a genetic sense, to future Martians what the small group of Homo sapiens who left Africa 70,000 years ago are to all non-African humanity today. The bottleneck through which an entire lineage passed, the progenitors of everything that came after. The sobering part is that if we send too few people, if the founding population is too small or too genetically similar, the colony's long-term viability could be undermined by inbreeding and genetic drift, regardless of how good the technology is. And if we use genetic engineering to modify those founders, we are not just changing their individual biology. We are making a choice about the genetic future of an entire lineage that will not have any say in those decisions. Think about that. Think about the responsibility of it. There's a concept in evolutionary biology called the founder effect, the loss of genetic variation that occurs when a new population is established by a very small number of individuals. Isolated populations founded by small groups often show reduced genetic diversity compared to their source population, and this reduced diversity can limit their adaptability to future challenges. If the founders of Mars are also genetically modified, The interplay between the artificial modifications and the reduced natural genetic variation creates a situation unlike anything in evolutionary history. A population with modified, non-random genetic variation but reduced random natural variation. How that plays out over generations is genuinely unknown. One idea that has been proposed to address the genetic diversity issue for Mars colonization is the use of a digital genetic bank. Sequencing the genomes of thousands of Earth humans and storing that information digitally on Mars, then using reproductive technologies to introduce genetic diversity as needed, essentially maintaining a virtual gene pool much larger than the physical population. This concept, sometimes called a digital ark for Mars, would use synthetic biology techniques to generate sperm, eggs, or entire embryos from stored genomic information, providing a reservoir of genetic diversity that could be drawn upon over centuries. The technology to do this doesn't currently exist at the scale required, but the component technologies, genome sequencing, artificial gamete generation, embryo cryopreservation, are all advancing. The digital genetic bank idea sits at the intersection of everything we've been discussing: genetic engineering, reproductive technology, population genetics, long-term sustainability, and the ethics of designing future generations. It is, simultaneously, one of the most scientifically creative and one of the most ethically fraught ideas in the Mars colonization conversation. As we begin to wind down, I want to take a moment to zoom out further than we have at any point in the story, to the deepest possible scale of time and meaning. Because there's a perspective on this whole conversation that only becomes visible from very far away, and it changes the emotional register of everything we've been discussing. For most of the history of life on Earth, which is to say for most of 4 billion years, the diversity of life on this planet has been the result of natural selection operating on random variation. Mutations happen by chance. The ones that confer survival advantages get passed on. The ones that don't. Evolution is blind, purposeless, and unimaginably slow. It has no goal. It has produced extraordinary things: consciousness, language, love, art, the capacity to understand the universe. But it produced them as byproducts of the simple, grinding mechanism of differential reproductive success. There was no intention, no design, no one who looked at the raw material of chemistry and said, "I will make a being that asks questions about its own existence." And yet here we are, asking questions about our own existence, and now beginning to answer them, in the most literal possible sense, by considering whether to rewrite the code from which we are made. If we do this, if we genuinely begin to design human beings for specific environments and specific purposes, we will be adding intentionality to evolution for the first time in the history of life on Earth. We will be taking the process of adaptation, which has always been random and retrospective, and making it deliberate and prospective. We will be looking at Mars and saying, we know what is needed to survive there, and we will build beings who have those qualities. That is a power of creation that no species in the history of this planet has ever had. It comes with consequences we cannot fully foresee. It comes with risks we cannot fully calculate. And it comes with a moral weight that is genuinely difficult to hold, because we cannot ask the people most affected by our choices, the future Martians themselves, what they want. We can only do our best to imagine what they would say if they could, and then try to honor that imagined voice in our decisions. What would they want? What would the first human born on Mars, 50 or 100 years from now, want their ancestors to have done? Would they want to have been engineered for their world, given every biological advantage we could provide? Or would they want to have been born as their ancestors were, human in the old sense, unmodified, carrying only the genes that 4 billion years of Earth evolution gave them, and adapting to their new world through medicine and technology and sheer determination? I don't know the answer. I'm not sure anyone does. But I believe the question is one of the most profound that our species has ever asked. There's a phrase that Carl Sagan used, which has become almost a cliché through overuse, but which deserves to be heard again in this context. We are the cosmos become conscious of itself. Stars formed and exploded, and their matter condensed into planets, and on one of those planets, chemistry became biology, and biology became minds capable of looking back at the stars that made them and understanding something of what they are. We are made of stardust, as Sagan said, but we are stardust that looks at itself in the mirror and wonders what it's capable of becoming. Mars is stardust too. The same supernovae that generated the iron in our blood generated the iron that makes Mars red. We are kin to that world in the most literal, atomic sense. And the question of whether to change ourselves to be able to live there is, at the deepest level, a question about whether we want to reunite those pieces of the cosmos. Whether we want to extend consciousness to another world, even at the cost of changing what that consciousness looks like. That is not a small question. It is perhaps the largest question that has ever been asked in human history. And the answer, whatever it turns out to be, will define not just the future of humanity, but the future of life in the solar system, perhaps in the galaxy. Before I leave you, I want to make sure you have a sense of the real human beings at the center of the story, not the abstract categories of scientists and ethicists and colonists, but the actual people who are thinking these thoughts and doing this work and carrying these burdens right now. There is Jennifer Doudna, sitting in her office at UC Berkeley, who has described publicly the troubling feeling of receiving a message from a colleague containing a photograph of a baby, with a note that the photograph was taken because she and her colleagues had developed CRISPR. She has said that she has nightmares about the misuse of the technology she helped create. She co-authored the Nobel Prize-winning work, and she spends significant time and energy on the governance and ethics questions. She started a committee called the Innovative Genomics Institute specifically to grapple with these questions. She is not naive about what she has made. She is carrying the weight of it actively. There is Christopher Mason, working at Weill Cornell, simultaneously excited about the biological possibilities of engineering humans for space and deeply aware of the ethical precipice. He has said that we have a responsibility to future generations to prepare them for the environments they will inhabit, and that refusing to use all available tools to do so is itself a moral choice with consequences. He has also emphasized that all of this work must be subject to the most rigorous public ethical debate. He holds both the excitement and the responsibility simultaneously. There is the anonymous group of researchers, many of them early career scientists in molecular biology and evolutionary genetics, who work in labs around the world on pieces of this puzzle, on CRISPR delivery mechanisms, on extremophile gene function, on human genetic variation in extreme environments. They often don't know exactly how their work fits into the bigger picture. They are doing science, piece by piece, paper by paper. But the cumulative effect of their work is the construction of a capability that will one day be transformative. And there are the future people, whoever they are, who will one day make the decision, or find it made for them, to be among the first to take this technology and apply it to the project of surviving on Mars. They are not born yet, some of them. They are children now, others. They are students in biology programs, dreaming of making a difference. They are the generation that will live with the consequences of the choices being made today in policy offices and ethics committees and government funding agencies. They deserve the most serious, most honest, most carefully considered debate that we can provide. The sky over Mars right now is empty of human presence. The only things we've sent there that still function are robotic explorers. The Perseverance rover, digging in the dust and looking for signs of ancient life. The Ingenuity helicopter, demonstrating that flight is possible in that thin atmosphere. The various orbiters mapping the planet from above. They are our ambassadors, our eyes and ears in a world we haven't yet been able to reach in person. They are remarkable, but they are not enough.